Imagine a microscopic enemy hiding in plain sight, really, lying dormant in the soil all around us. And it's capable of turning just a simple cut into this devastating neurological crisis. It's a disease we often think about with humans, maybe horses. But today, on the deep dive, we're looking at its surprising impact on two quite unique animal families, camelids and cervids. We are talking about tetanus. And we've got a stack of veterinary reviews, research excerpts here. Our mission today is really to unpack the critical nuances of this persistent threat. In camels, llamas, alpacas, deer, elk, you'll find out why it can be so hard to spot, especially in certain populations, what actually happens when it takes hold, and honestly, some unexpected critical gaps in how we protect these animals. Get ready to maybe rethink some basic assumptions about animal health. Okay, with that chilling premise set, let's really pull back the curtain. Let's understand this unseen enemy beyond just the name tetanus, what's actually happening down at the cellular level when it strikes.
Right. So tetanus is a severe neurological disease, often fatal. But the really crucial thing here, it's not the bacterium itself, Clostridium tetani, that causes the direct problem. It's actually a potent neurotoxin it produces called tetanospasmin. Now, the bacterium, it's what we call gram-positive, that just describes its cell wall, and critically, it's anaerobic.
Anaerobic, meaning it hates oxygen.
Exactly. It absolutely thrives in environments completely devoid of oxygen. It also forms these incredibly resilient spores. Under a microscope, they actually give it this characteristic drumstick appearance. It's quite distinctive.
Wow. Drumstick spores. And you said ubiquitous. How widespread are we talking?
Truly remarkable. They're found globally, in soil, dust, even in the intestinal tracts and feces of many animals. Think horses, cattle, and yes, even humans carry them sometimes. They're like little environmental survivalists. They can persist for months, even years. They shrug off heat, drying. Many common disinfectants, they're essentially just, waiting for the right opportunity.
So it's literally everywhere, like these silent microscopic landmines just waiting. But for these super tough spores to actually cause disease, they need a very specific kind of environment once they get inside an animal, right? They can't just germinate anywhere.
Precisely, you got it. The spores themselves, they're biologically inert in healthy oxygenated tissue. Think of them like tiny inactive seeds. They only germinate or kind of sprout into the active toxin-producing bacteria when they land in an anaerobic environment, a place lacking oxygen.
Okay, so where do they find that? What creates those conditions?
Typically, it's things like deep puncture wounds. Stepping on a nail is the classic human example. Or crush injuries, surgical sites sometimes, wounds that have foreign bodies in them, or necrotic that's dead tissue. Anywhere oxygen levels are significantly reduced. Common entry points we see in animals. Often puncture wounds to the foot, castration sites, umbilical stumps in newborns, even trauma after giving birth. Basically, any place where oxygen is cut off is like an open invitation.
Right. And once those spores do germinate and start pumping out that tetanospasmin, that's when the real trouble starts. How does this neurotoxin actually wreak such havoc? What's its strategy inside the nervous system?
That's where it gets truly insidious and quite sophisticated, really. The tetanospasmin neurotoxin, it doesn't just stay put at the wound. It disseminates, it travels, it gets into the lymphatic system, the bloodstream, and it can be taken up directly by peripheral nerves right near the wound. Then it literally hitches a ride. It travels via what we call retrograde axonal transport.
Retrograde, going backward.
Exactly. Backwards up the nerve pathways, heading straight for the central nervous system, specifically targeting the spinal cord and the brainstem. And this backward ride means that wounds closer to the CNS, say on the neck or head, they're often associated with a shorter incubation period, usually 10 to 26 days. And often that means much more severe disease.
Okay, reaches the CNS. Then what? What's the specific damage it does there?
Right, so upon arrival, the toxin performs its terrifyingly specific action. It irreversibly blocks the release of inhibitory neurotransmitters. These are the chemical messengers like GABA, gamma, amino, butyric acid, and glycine. Their job is basically to tell muscles to relax, to calm down. By blocking them, the toxin effectively removes the off switch for the motor neurons.
So the off switch is gone, just gone. And the muscles are just firing constantly, like being stuck in overdrive.
Precisely. Stuck in overdrive is a good way to think about it. Without that constant inhibitory signal telling them to chill out, the motor neurons become hyperexcitable. They fire continuously without any regulation. Imagine like your car's accelerator pedal is jammed to the floor and the brake pedal just doesn't work.
Okay, yeah. That's a scary image.
It leads to profound, generalized muscular rigidity. We call it hypertonia, extreme muscle tension, and violent, painful reflex spasms. These can be triggered by even minor things, a sudden noise, a gentle touch, even a flash of light. The classic signs are pretty distinct. You see trismus, that's lockjaw, the jaw muscles just seize up, a rigid sawhorse stance where the animal's legs are stiff and splayed out, difficulty swallowing dysphagia, which leads to starvation, dehydration, and often prolapse of the third eyelid. You'll see that membrane covering part of the eye.
And you said irreversible, the toxins effect.
The toxin's action on those specific nerve endings, yes, it's irreversible. So recovery, if it happens at all, depends on the body actually sprouting entirely new nerve terminals to bypass the damaged ones. And that's a process that can take weeks, sometimes many months. It's a long, really arduous fight for the animal.
Wow. That paints a vivid and, frankly, pretty chilling picture of how this disease works. Okay, so that's the general mechanism. Now, let's zoom in on our specific groups today. Camelids and cervids. Are they equally susceptible? Or do we see different risk factors, maybe different ways the disease shows up in them?
That's a great question. And the answer is, well, it's complicated. Both families are susceptible, yes. But the reported incidence, how often we actually see it confirmed, and the diagnostic challenges, they vary significantly between them. It makes their situation quite unique.
Okay, let's start with camelids then. So camels, llamas, alpacas.
Right, camelids. Historically, there was this idea that dromedary camels, the one-humped kind, had some natural resistance to tetanus, mainly because, well, reporting cases were pretty low. But more modern reports, recent studies, they've decisively confirmed fatal cases. It shows there's full susceptibility across all the species. Old World camels, New World llamas, and alpacas. What probably looked like natural immunity before was maybe more likely a case of it being a silent killer. Perhaps just not noticed or diagnosed due to less veterinary surveillance in some areas.
Interesting. So what are the big risk factors for them?
For camelids, the risk seems strongly linked to human management practices. Things we do. Post-castration wounds, especially in young male dromedary camels, that's a notable cause. These are often open wounds that, unfortunately, create that perfect, deep, oxygen-poor environment for the spores to germinate. There are even detailed case studies where C. tetani was isolated directly from a castrated camel, and they traced the source back to contaminated soil in a nearby horse paddock.
Wow, so it shows just how easily it can spread between areas or even species indirectly.
Exactly. It highlights how pervasive the threat is. Other risks include things like ear tagging procedures and umbilical stumps in newborn crias at the babies. Basically, any procedure that creates a potential entry point for the bacteria.
And clinically, how does it look in them? Same as other animals.
Mostly, yes. The presentation is largely consistent with other susceptible species. Generalized tetanus is the most common form we see. But, interestingly, localized forms can occur. There was an experimental study done in guanacos. It's a wild South American camelid. They found that injecting the toxin in the neck region produced wryneck, or torticollis. That's a localized muscle rigidity, a kind of twisted neck posture. And that has been seen in clinical dromedary cases too. But fascinatingly, when they injected the toxin in a hind limb in that study, it only caused localized spasms in that specific limb. And the animal actually recovered from that.
Really, just localized. That suggests the wound site might really influence the outcome.
It certainly seems to. It suggests the site of entry can influence not just the incubation period, but maybe the form and overall severity of the disease. It's not always a whole-body catastrophe immediately.
That's a really surprising detail, that wryneck, and the possibility of localized forms. It shows how varied it can be. Okay, what about cervids then? Deer, elk, moose. I mean, I imagine diagnosing something like this in a wild, free-ranging deer must be incredibly difficult, maybe completely overshadowed by other diseases people are looking for.
You've absolutely hit on a crucial point there. For cervids, particularly in wild populations, the confirmed incidence of tetanus is strikingly low, really low. But this immediately raises a big question. Is that apparent rarity because they have some true biological resistance? Or is it mostly down to significant diagnostic challenges and just limitations in our surveillance? Honestly, we just don't have enough solid data to be certain. It's a bit of a black box.
So what are the risks for them? Does it differ between, say, farm deer and wild deer?
It does, somewhat. For farm deer, there's definitely an elevated risk from injuries. Things that happen during handling in yards, during transport, maybe from fighting with each other, stags fighting fiercely during the rut, that can lead to deep puncture wounds, perfect tetanus entry points. And the removal of velvet antler, that's a common management procedure on deer farms. That also creates potential entry sites.
Makes sense. And wild deer.
Wild deer, well, they're subject to more natural traumas. Puncture wounds from sparring with other deer, injuries from predators, lacerations from things like fences or other environmental hazards. We also see potential for increased injury risk across a whole population after severe weather. Think hurricanes, tornadoes, all that flying debris can cause widespread trauma.
Right. And the diagnostic challenge you mentioned, how big a problem is that for wild cervids?
It's substantial, absolutely substantial. You have to remember the primary neurological diseases everyone is concerned about in wild North American cervids are chronic wasting disease, CWD, and rabies. Both of those have extensive mandatory surveillance programs, huge amounts of testing.
So everyone's looking for CWD and rabies.
Exactly. So when a wildlife official or biologist sees a wild deer or elk showing any neurological signs, stumbling, acting weird, weakness, the default suspicion, quite understandably, is often CWD or rabies first. Now think about the signs of tetanus. That spastic paralysis, the rigidity. From a distance, couldn't that easily be misinterpreted as the staggering gait or the incoordination we associate with CWD or rabies?
Yeah, I can absolutely see that, especially if you're just observing from afar.
And here's the critical twist. Diagnosing tetanus is fundamentally clinical. It relies heavily on recognizing those characteristic signs, plus having a history of a recent wound. Now, how likely are you to get that history for a free-ranging wild animal?
Almost impossible, I'd imagine.
Pretty much. Imagine trying to track a specific deer in a huge forest or prairie, and then trying to find a small puncture wound, maybe from a thorn or a minor fight. That happened weeks ago. It's just not feasible. We're talking about animals that are masters of hiding injury, living in environments where close, sustained observation is incredibly difficult.
So without that key piece, the wound history tetanus just stays hidden.
It stays hidden in plain sight. And with that intense necessary focus on CWD and rabies testing, it's highly plausible that actual cases of tetanus are either being misdiagnosed as something else, or, maybe more likely, the possibility of tetanus just isn't even investigated. They do the CWD rabies test, it's negative, and that's the end of it. Plus, animals that die in remote areas, they're likely scavenged long before anyone finds them for sampling. So you put all these factors together, it creates this significant diagnostic bias. It strongly suggests the true incidence of tetanus in wild cervids is probably much higher than the official numbers show. It's a genuine blind spot in wildlife health.
That really does paint a bleak picture for diagnosis, especially for wild cervids, but even for managed camelids where maybe a wound gets missed. So if it is suspected, despite how hard it is to spot, how do vets actually diagnose it? And then what's the treatment outlook? Is there much hope for recovery once those neurological signs appear?
You're right, it's a real puzzle for the veterinarian. Diagnosis relies almost exclusively on recognizing those characteristic clinical signs we talked about, the lockjaw, sawhorse stance, spasms, ideally combined with any available history of a recent wound. Unfortunately, lab tests aren't much help for confirming it in a live animal, what we call antemortem confirmation. You can theoretically try to culture the C. tetani bacteria from the wound, but it's often difficult. The bacteria might only be there in small numbers, and remember, the real issue is the toxin that's already spread. It's often unrewarding.
So no easy blood test or anything?
No, not for confirmation in a living animal showing signs. Definitive diagnosis usually happens after death, post-mortem. That involves isolating the organism from the wound site, if possible, and then confirming toxin production using a specific test, usually a mouse inoculation bioassay. That tells you the toxin was indeed present, but of course that's too late for the animal itself.
Right, so differentiating it from other neurological diseases must be key.
Absolutely paramount. Given how much the signs can overlap, differential diagnosis is crucial. A thorough clinical exam, gathering whatever history you can, however limited, it's essential. Vets have to systematically rule out other conditions. Rabies, with aggression, paralysis, maybe drooling. Listeriosis often causes one-sided brainstem signs like circling or a head tilt. Polioencephalomalacia, PEM, that can cause central blindness, a stargazing posture. CWD with its weight loss and behavioral changes. Meningeal worm, causing asymmetric paralysis. Various viral encephalities. Tetanus has to be on that list of possibilities, even if you can't find an obvious wound, precisely because those wounds are so easily missed.
Wow, that's a real medical detective story for the vet. So many possibilities. But let's say they do figure it out, they make the diagnosis. Can these animals actually recover? What does treatment even look like? Is it often successful?
Well, the unfortunate reality is that treating clinical tetanus is incredibly intensive. It requires significant resources, time, money, specialized care. And sadly, it's often unsuccessful, particularly if treatment starts after severe signs have already developed. The prognosis is generally considered guarded to poor. We estimate the case fatality rate approaches 80% in animals that haven't been vaccinated. It truly is a race against that potent toxin, which is likely already bonded to nerve cells.
80%.
Yeah.
That's incredibly high.
Yeah.
So what does the treatment involve? What are they trying to do?
It's a multi-pronged approach, trying to fight it on several fronts at once. First, the absolute immediate priority is to neutralize any unbound toxin that's still circulating in the body before it can reach the CNS. For this, you give tetanus antitoxin, or TET-T, immediately. This antitoxin essentially grabs onto any free-floating tetanosspasmin and disarms it. Doses for camelids and cervids. They're largely empirical, based on experience, not specific research for these species. They can range from, say, 10,000 up to 100,000 international units, or even more for treatment. It's a high-stakes, very time-sensitive intervention.
Okay, neutralize the free toxin. What else?
Second, you have to eliminate the source of any new toxin being produced. That means the wound needs thorough debridement. You meticulously remove all the dead, necrotic tissue and any foreign material. The goal is to create an aerobic environment, get oxygen back in there, which inhibits the C. tetani bacteria from growing and making more toxin. You also give high doses of antibiotics, typically procaine penicillin G, to kill off any remaining active bacteria at the site. Stop the factory.
Makes sense. Stop the toxin production.
Yeah.
What's the third part?
Third, and arguably the most critical component for survival, is intensive, supportive, and nursing care. This is hugely demanding. The animal has to be kept in a dark, quiet, really well-padded stall. You want to minimize any external stimuli, noise, light, touch that could trigger those severe, potentially life-threatening spasms. Sedatives like diazepam and muscle relaxants are crucial. They help control the rigidity, reduce the convulsions, maybe make the animal a bit more comfortable. And nutritional and fluid support are absolutely essential. Remember, these animals often can't eat or drink because of the lockjaw and difficulty swallowing. You might need IV fluids, maybe even tube feeding.
That sounds incredibly intensive. And you said despite all that.
Despite all these intensive efforts and really dedicated care, most of the reported clinical cases, especially in dromedary camels, for instance, have sadly been fatal. Or they reach a point where euthanasia is the only humane option because the disease is just progressing relentlessly. It's a tough battle.
It really sounds like an incredibly challenging, often heartbreaking situation for everyone involved. So, given how devastating it is, how hard it is to treat, prevention seems like it must be the only real weapon we have. We know there are highly effective vaccines for humans and horses. It seems like a straightforward solution for them. So for camelids and cervids, surely it's a similar story, a clear path to protection.
You would absolutely hope so. That would be the logical conclusion. But this is precisely where we hit a really significant and I'd say critical gap in veterinary medicine when it comes to these specific species. The stark reality is there are no tetanus toxoid vaccines officially licensed for use in either camelids or cervids. None.
Wait, none at all for animals we know are susceptible?
None specifically licensed for them. It's a profound vulnerability in our ability to protect these animals, especially considering the severity of the disease.
So how do people try to prevent it then?
Well, prevention relies entirely on the off-label use of multivalent clostridial vaccines. These are typically the 5-in-1 or 7-in-1 type vaccines, combinations that protect against several clostridial diseases, including tetanus, but they are actually licensed for other livestock, primarily sheep and cattle.
Off-label use. What does that mean exactly in practice?
It means that while this practice is common and it's widely recommended by veterinarians who work with these species, it also means that the efficacy, how well it works, and the safety of that vaccine are not guaranteed by the manufacturer for use in camelids or cervids. And crucially, the legal responsibility for any issues arising from its use rests entirely with the prescribing veterinarian, not the drug company.
Okay, that sounds risky. And do we even know if these off-label vaccines actually work well in them?
That's the other major concern. The immune response to these off-label vaccines can be quite variable and potentially insufficient. There was a study done in red deer, for example. It showed that while a 5-in-1 clostridial vaccine did induce an antibody response against tetanus, the peak antibody levels, the measure of protective antibodies in the blood, were only about 10 to 20% of the levels seen in sheep and cattle, the species the vaccine was actually designed for.
Only 10 to 20%? That doesn't sound very protective.
It certainly raises serious questions. And furthermore, the response in those deer was of shorter duration. It didn't last as long. So this makes us ask, Is the immune response we're generating actually strong enough to be truly protective in these species? And how often do we really need to give booster shots to maintain even that lower level of immunity? We're essentially operating with educated guesses based on protocols for other animals.
So we're basically navigating this really critical health threat with educated guesses, using vaccines made for sheep and cows, and just hoping they provide enough protection for llamas or elk. That feels like a real vulnerability, especially, like you said, with increasing numbers of farmed camelids and cervids or valuable wildlife conservation programs.
It is, absolutely. And it highlights that stark reality. We lack species-specific, evidence-based vaccination protocols for these animals.
Yeah.
It really does feel like we're kind of operating in the dark a bit when it comes to guaranteeing truly protective immunity against tetanus for them.
So pulling this all together for you, our listener, what does this all mean? We've gone from this microscopic spore that's just everywhere in the environment, impossible to avoid, to the devastating way its toxin hijacks the nervous system.
Yeah.
And through the huge challenges of spotting the disease, especially in the wild, and the often unrewarding fight to treat it. What really stands out to you from this whole deep dive into tetanus and camelids and cervids?
For me, a few key things. First, the sheer ubiquity of Clostridium attoni. It's everywhere. We can't escape exposure, which means prevention has to be our primary strategy. It's really the only viable one. Second, the incredibly high mortality rate, even with intensive, costly treatment. That just underscores the urgency of prevention. We also learned that for both camelids and farmed cervids, human management practices are often the primary risk factor creating those wound opportunities. While in wild cervids, natural trauma, maybe severe weather events, represent this largely unquantified and I strongly suspect underestimated threat.
And that critical point about underdiagnosis in the wild.
Yes, we absolutely can't forget that. The probable underdiagnosis in wild cervids, that significant blind spot, largely because everyone's understandably focused on CWD and rabies surveillance. Tetanus is likely hiding there, in plain sight, more often than we realize.
Absolutely. And the stark reality you just highlighted, that we simply lack species-specific evidence-based vaccination protocols. We're relying on hope and extrapolation from other species for their protection. It feels like a profound ethical and practical gap.
Precisely. And this points directly to a pressing need. We need controlled, targeted research. We need studies designed specifically to determine the immunogenicity, how well these vaccines actually trigger an immune response, and the real efficacy of existing commercial tetanus toxoids in various camelid and cervid species. We need to figure out what level of antibody actually counts as protective for them, and then define optimal vaccination schedules based on solid science for these animals, not just borrowing from sheep and cattle protocols. We need to bridge this knowledge gap.
And that really leads us to our final provocative thought. Something for you to mull over after listening today. Considering how widespread this bacterium is, how severe and often fatal the disease is, and the current challenges, the uncertainties surrounding these off-label vaccination approaches, what responsibility do we have? As humans, as caretakers of these animals, whether they're on our farms and zoos or simply sharing the wild landscapes with us, what responsibility do we have to invest in the specific research needed? To develop targeted, truly evidence-based protections for these unique and increasingly important animal populations. It's definitely something worth pondering.